Hi everyone, I'm Jil Meier, and I'm a postdoc in the brain simulation section in Berlin, where I work with Professor Petra Ritter on the virtual brain. I've done so for the last 2 years, and I'm especially interested in deep brain stimulation. So I hope in the next half an hour or so, I can make you as enthusiastic about it, and I can help you understand the basic principles of our simulation and of our generative models. All right, for that, let's get started. Okay, I will first give you a little bit of an introduction of what's possible with the virtual brain, how does it work, and then we will slowly move on to deep brain stimulation, a certain format of neuromodulation. And then we will, in the end, hopefully, come to simulating it ourselves on your computers. All right, so let's see. We want to stimulate a certain region of the brain, and then we want to see that stimulus propagate around the network of the brain through different regions, altering brain dynamics. That's the ultimate goal here. So the virtual brain, maybe you've heard about it, maybe you did not hear about it so far, but the virtual brain is literally a virtual brain. So it's a neuroinformatics platform where you can simulate brain activity. You can simulate brain activity on different scales. You can simulate neurons. You can simulate also whole brain dynamics. And mostly it's focusing on regional activity. So this picture gives you a slight overview in a short version how the virtual brain works. You can see that as an input, we need a real brain, an empirical one, or at least data of it. So most often the data that's used to input to the virtual brain is individual MRI images. T1 or diffusion weighted imaging, both would be best. And also we like to have functional data of an individual to make sure that what comes out of the brain, out of the virtual brain, the functional network, is actually similar to the empirical data of the individual subject. So the ingredients are data as an input, and then simulated data as an output. And what you need to determine and what you can choose inside the virtual brain is a model. How would you like your brain to work? So in that model, most often we have population models. That means that we don't simulate each individual neuron, but we simulate the average activity of a whole population of neurons. So here, for example, each brain region is given two populations, one excitatory neuronal population and one inhibitory one. And they're influencing each other, and all of this will be described in what we call a neural mass model. And out of this comes a firing rate, some synaptic activity that's then, for example, translated into bold signals. And then we can compare that again with, is it how it looks empirically? And people have done that for quite a long time now. The virtual brain was born in 2009, and then they came closer and closer to the reality. But still, it's of course a model, it's an abstraction of what really happens in the brain. So if that was maybe too fast for you, let's split it into input and output of the virtual brain. So the input to the virtual brain, what you would need to simulate your individual brain, would be an MRI. MRI image of best diffusion tensor imaging weighted image with choosing a certain parcellation, which then gives you a matrix. This matrix is nothing else than a description of all the tracts in the brain, the tract's length, and also the thickness of these tracts. So for example, number of streamlines, then you would like to know how long are the streamlines between each pair of regions and how many tracts are there. And then something happens inside the virtual brain. We'll come to that magic in the next slide. And then what you get out is actually local field potential. So in our case, for later already, we can see that you can get out local field potentials right around the electrode that's placed inside the brain of patients. You can get a time series of different modalities. For example, the virtual brain will output you an EEG time series or an MEG time series. or also a bold series. You can choose yourself and it's all implemented and ready for you to be used. All you need to change is one line of code for that. All right, so what's inside the virtual brain? How does it look like? I've said already a neural mass model. So here the virtual brain makes use of this principle, what is often coming from complex systems theory, where you have a mean field theory, meaning that instead of simulating the activity of every single bird that's flying through the sky here, we focus on the average bird, and still we will be able to trace the birds in the sky. So that's what we do with neurons. We basically say, okay, we will not focus on a single neuron and then compute its activity 100 million times for different neurons, but we will summarize them. And we will say in each region of the virtual brain, we have, for example, one inhibitory, one excitatory population. And these populations are in each region the same for the virtual brain. And then these populations have a certain rule how their activity is generated, and that's described with these couple of differential equations here. What they basically give you is a description of a certain state variable, for example, the membrane potential for that whole region for a time point, and then the equation gives you the membrane potential for the next time point. So these equations are just a description, mathematical description of how over time the activity of a single brain region will evolve. So once we have this model working for every brain region, it will give us the activity of all the brain regions. But then what we also do, because our brain are not just single regions placed in space, but they're connected. And that's where the input data of the structural network, the DTI or T1, comes in. And we connect those regions via a link. This link is this streamline. And we will also have a variable called global coupling that we will be able to scale the link a bit higher or lower. So basically we couple all these regions together. And then already it will give us activity of the whole brain, and that will give us the virtual brain. So what has been researched so far with the virtual brain, or we abbreviate it TVB? You will hear all about it from Professor Jil in a later talk today, where the virtual brain is currently used in a clinical trial for epilepsy patients, where they try together with the surgeons to come up with the best area to basically cut out of the brain using the patient's virtual brain. So it's used as an advisory tool to neurosurgeons already. Also, there have been multiple retrospective studies that show that the virtual brain could in principle also be used to determine how much of the tumor tissue should be resected. And also a study where they were already trying out virtual therapies, like in this case, the drug memantine in Alzheimer's patients, to come up with the empirical response of this pharmacologic intervention and try to basically give a therapy here to a virtual patient first, see the effect, and then this could be used again as an advisory tool for neurologists or neurosurgeons. So lots of cool things have been happening with the virtual brain already. And when you want to know more specifically what's basically the core of the virtual brain, it's this one equation. So as you can see, there is not a lot of magic here, let's say. It's one equation. What's this equation? It's a differential equation. And this is just the differential operator telling you what's the next time point of x. So we give in x and we get out of this whole equation the activity of x at the next time point. So what is in there? We don't just give x, but we give x could be, for example, a membrane potential, a firing rate of a region. So something we can measure right now in the state variable. And then we give it into our neural mass model. And our neural mass model will tell us what happens next. The these can be, as I said, firing rates, field potential, some state variable that you are interested in, and that is described by these differential equations. Then I already said we will hook up the regions together, and by that we will also scale the strengths of their connection with the factor G, which is a global coupling scaling factor. It's just a number. And then the structural connectivity also needs to be in there, right? It will determine which regions are actually connected with each other. And then we also have a certain time delay. This is why we need the track length, because even though a connection may be very strong, if it's very long, we need to kind of take that into consideration and say, if we have a longer connection, there will be a certain time delay that the activity of one region will arrive at the other one. So this can also be simulated inside the virtual brain. And then there's also a local connectivity. That's not for today. That's you can complicate the virtual brain as much as you like. There are so many features you can plug in and things that people have done with it. Not for today, but this is basically how much are you influenced, not through the white matter fiber tracts by other regions, but through the just spatial proximity of being close to another one. So if I'm a region and I'm very close to another region, then I will be influenced by that activity as well, even though we may not have a very strong fiber tract in between us. So you can have different kinds of connectivity. Let's sum it like this. And this is the injected input, which is something that we will play with today. It's, for example, inputting a stimulation. And you also have noise, as always. So how will we use this equation now to go using the virtual brain from a diseased brain towards a healthy target brain in this schematic now 3D space of brain states? We will basically try it out for an individual patient inside the virtual brain, play with the different parameters of different stimulation techniques, use different stimulation techniques, use medication, and then hopefully the vision is that we will make the dynamics of the patient again closer to the healthy dynamics. This just to give you a picture of what, the community of the virtual brain is working towards. But there are many, many different approaches, and it's an open source software, so it's been used for so many different things. If you're interested, just check out the website, thevirtualbrain.org, or maybe you've already checked it out and already downloaded it, then you're ready to go in a second for our hands-on session. But first, let's move towards now the specific motivation why I said that I want to do this tutorial. It's because we want to simulate deep brain stimulation, or short DBS. So the motivation behind is actually that we want to predict optimal targets for DBS. What's DBS? It's a surgical procedure where an electrode, or basically in principle 2 electrodes, like little chopsticks, are placed inside a patient's brain that then give out some stimulation. And this stimulation will change the dynamics of that brain and will often lead to symptom improvement in patients. So this is now focusing on using the virtual brain and deep brain stimulation to move from the diseased brain to the healthy target brain. And why could the virtual brain assist here? Because these are, for example, different 20 patients taken together, and their electrodes are mapped after the surgery. Where were they placed? And you can see they were placed kind of all over the place. And this means that the specific placement of the electrode actually can vary. And this is good if it's helping the patients, but it's bad if we don't know what we're doing there. So often what has been then found by Horn et al. Is actually that once you place the electrode and it stimulates some red fiber tracts here, then this will lead to improvement for the Parkinson's disease patient. But when you place the electrode inside the brain and it touches upon these blue fibers, activating them, this is actually clinical worsening for the patient. So you, by all means, want to avoid that. So you can see already it's on a millimeter scale, a very important decision where to place the electrode. And individually, your tracts can vary. Individually, your dynamics can vary. So this needs to be tailored more specifically for the individual patient, which could then lead in helping and advising neurosurgeons on where to place it. So the underlying mechanism is indeed not yet completely understood. What does deep brain stimulation? But it was found that it helps quite a lot of patients in different diseases. So we thought modeling could help here. And there are DBS models on different scales, actually. You can see that people simulated deep brain stimulation on the level of single neurons. And they were looking at what does the stimulus do to a single neuron. And on the other hand, people were already looking into whole brain dynamics of deep brain stimulation. So what does this stimulus actually cause also in areas being located further away from the stimulation area? So we built a multi-scale model to bridge over those different scales. And I will explain that in a second. This multi-scale means basically some regions, the regions around the electrode, were simulated on a very high level of detail, including spikes, so real single neurons and their spike trains, and other areas being further away. They were simulated with neural mass models, looking at them from a bit further away, like looking at them on the population level. So what's the vision here? We would wish that the Parkinson's disease patient comes in, we get his structural MRI and also his functional data. It could be EEG, MEG, fMRI, LFP signals, whatever you prefer. And we use that as an input to the virtual brain and validate it back and forth to set all the parameters in the correct manner, such as we have a virtual representation of the brain activity of that patient. And what can we do with that then? We want to optimize the placement of the electrode, but also the settings of the electrode using high performance clusters that we have available. Then we can test all the different stimulation settings. Instead, in the clinic, it actually can take up to a couple of months for the patient, together with his neurologist, to get the setting correctly for the deep brain stimulation. So you can play with different frequencies, you can play with different placements, you can play with different amplitudes of the stimulation and see what would be ideally the best outcome for the patient from our dynamic systems perspective. And with that, you would personalize the treatment of the patient. So when we used our multi-scale model, we used all the regions that were around the electrode to give out spikes. These are raster plots. So every dot is really over time a single spike of a single neuron. And at the same time, all other regions, something like this came out. It's an average activity of the firing rate of this region. So we then placed virtually stimuli here. And the paper about this was just accepted actually in experimental neurology, where we showed that using this model, and using deep brain stimulation inside the model, it will actually look like this. This is from a patient, Parkinson's disease patient, his resting state brain activity. Now the electrode is placed, and you can see that the colors keep on changing also in regions much further away. This is a firing activity of those regions. So you can see with this continuous stimulus here that it travels and influences the dynamics of the whole brain. Grades go here to our media artists who made this nice video of it, of our results of this study. All right, so before we watch it again, let's look at the results in plots. So when we looked at the different regions around the electrode, what was interesting to us is that the thalamus activity actually was increased by virtual deep brain stimulation. And you can see here that the control has high thalamic activity, and Parkinson patients are known to have low thalamic activity. And with the virtual deep brain stimulation, we could already see that this will increase thalamic activity for the patient in our modeling world, let's say. And then we could not only see these spikes and these firing rates of the regions around the electrode, but also we could enlarge our view and have this view on the cortical regions and what happens to also regions much further away when we stimulate this tiny STN or GPI nuclei. So you can see that when we have the resting state from the patient and the control, that for example, the pre-central gyrus, post-central gyrus area, had actually lower activity here in the patient than in the control. But when we switched on the electrode, you can see that this area turned red or even black with different stimulation patterns, bringing the activity of that region back to the healthy activation level. Also with other regions here, for example, you can see the middle frontal gyrus. Gyrus is actually known from fMRI studies where they switch on the electrode, that this region plays an important role as well, even though it's much further away from where the electrode is placed inside the basal ganglia. But it's the biggest shift here in between DBS on and off stimulation scenarios. So you can see that this already brought us some insight, even though this was one control and one healthy patient. Feel free to have a look at the paper if you're interested, contact us for sure. And this is now coming towards the hands-on. So how is it actually looking, the virtual brain? The TVB software has a graphical user interface that you can see being recorded here. So you can just play with the graphical user interface if you don't like programming too much, or if you just want to get a nice video of brain activity, like here, feel free to start with the GUI. Here, actually, we have only simulated the basal ganglia with the GUI, and we stimulated the subthalamic nucleus, which is one of the targets used for brain stimulation in Parkinson's disease patients. And you can see how that one stimulus propagates through other regions, impacting their activity as well, and how it kind of spreads throughout the network. All right. So we have seen quite a lot now on basically how we can simulate this therapy of deep brain stimulation inside the virtual brain. And now it's basically up to you to do it. So I hope you've received the information, or you can look it up on the website, how to install the virtual brain. You can either download one of the folder TVB distribution from the website. That's maybe easiest, but you need probably good internet for that. Or you can also get an account at eBrains. Then you just have to request an account there, and actually within 1 or 2 working days, you often get the eBrains account. And then you have access to also simulating it via the browser, the graphical user interface, as well as the programming interface. Or you can just install it locally with pip and different kinds. So they are actually, or run it through a Docker. So whatever suits you to make it run locally or even in the browser. The version in the browser is great because you don't need a great computer. You can use the supercomputers from, for example, the Jülich Center in Germany as a backup. And that will make your simulations run faster, actually. So what will we actually do? So we will put our hands on the virtual brain and simulate it. And we will hopefully in the end get these nice plots ourselves. So we will define a stimulus and visualize it. We will apply a stimulus to a certain region based on a region-based simulation, one I just explained. And then we will visualize the results and see how it spreads around the stimulated area, also to other areas. So basically making our own video, but it's not yet playing. It will be actually just time snapshots, let's say. All right, so let's see how we can do it. And if you have not installed the virtual brain or you had problems with it, don't panic, just lean back and have a look now how I do it. I will explain it step by step, and later on you can do it at home and try to retrace the steps. So you should have the Jupyter notebook that I'm seeing here as well now. And this Jupyter notebook, let's say we can make it larger, is now setting up the virtual brain. So let me actually run it step by step and go through with it with you. So we are setting up the virtual brain. You can uncomment these lines, pip install, etc., etc. If you really have not yet installed the virtual brain on your local computer and you still need to do it, I commented them here because this actually makes it faster, of course. So let's run them all. All right, so back to, so he's importing everything, and then we are actually defining the stimulus, and for that we first have to give input to the virtual brain. The virtual brain comes with a couple of default connectivities, so with connectones already prepared for you. One of them is called connectivity 192, which is 192 because it's 192 regions, and we will use that one. So it's importing the connectivity with this command, and then we need to define where spatially will the stimulus be placed. And this is in a variable called weighting. We first make it all zeros vector, so the stimulus is nowhere. And then we will choose region 12 arbitrarily now, because this is a toy model, right? And give it a certain stimulus of 0.1 weighting. We don't know yet, so this is just spatially. So 0.1 of the stimulus will be placed there. You can also choose multiple regions and give them all 0.1. So this is still in the toy world. These are not real voltages or something. So you just place it and decide which regions. You can play around with it. You can place it at different regions and see if actually the pattern of the propagation of the stimulus changes. The temporal profile of the stimulus is also important. We chose what's already inside TVB. It's a pulse train. So really just a stimulus giving pulses. It will start at 1,500 milliseconds. And it will have a gap of 50 milliseconds, and it will last for 1,000 milliseconds. Okay, so now we need to define that stimulus. That's often in TVB that we put it all together in this stimulus class, and we visualize it. For that, we first configure the stimulus and then visualize it. You can see that I chose here one region to be stimulated after 1,500 milliseconds. So now we will set up the simulation. And actually these lines are all it takes to set up a simulation in the virtual brain. So you can choose a model, a neural math model, which is here the generic 2D oscillator. You can give it some parameters if you don't like the default ones. You have to give it some connectivity. You give a coupling. That's actually the G, or global coupling, that we talked about earlier. You need an integrator. That's just a function telling you how the equation, the differential equation, will be integrated. And then we will monitor it. So here we will just look at the temporal average activity of firing rate. You can have different monitors here, EEG or MEG or BOLT, for example. Everything just needs to change this line. And we give it a stimulus. And the simulation length is, it's actually not exactly 1 minute, approximately. We need to configure, and this line executes the simulation, giving you the time where it has recorded activity and the data it has recorded in an array. Then we can look at it and plot a figure. And this figure here now nicely shows as a red line the average activation level of all the different regions in the virtual brain. And you can see in black single activations. So you can see that some regions are quite strongly impacted by our stimulus. That's a pulsing stimulus coming back every 1,000 milliseconds again and again, right? So this is not really a nice visualization. We cannot see which region does what and how does the stimulus exactly propagate. So let's visualize it. And here the credits of this code go to John Griffiths, and you just execute this, defines basically a function how to visualize our activity time series on the brain. And we will need to import a cortical surface. That's basically just a mapping telling us which, so for every region, telling us what are the points that I have to color on the surface. That already comes with TVB as well. And then we will plot it. So this function here now plots what we've just times after 1,500 millisecond after the onset of the stimulus. And it plots the regional activity. So when a region is darker here, it means it has a high level of firing rate. All right, so we can see that it propagates. The most of the stimulus stays in region 12 that we actually just chose as stimulus beginning, but you can see that other regions much further away also react. All right, this is really nice. If that worked for you locally, it's even nicer. If not yet, we can get there. Just contact me as well. All right, so we can now stimulate a different area. So how will that actually differ in our code? And for that, all we need to do is go back to the line where we had 12, right? And maybe put 90, or let's say 30. And now let's actually just run everything again and see if we see a different pattern. All right, it's simulating now. And of course, depending on your local computer, this may take longer or shorter. It's giving some warnings we can ignore for now. And here, as you can see, I've chosen an area now in the temporal lobe, and it propagates from the right hemisphere all the way to the left hemisphere as well. So it has a huge influence on the whole brain activity. So you can play with that, test out different regions, test out your favorite regions, etc., etc. Or you can change the parameters of the stimulus, and we can see if that changes anything to our pattern. So we, for example, have here multiple different stimuli with different time courses. That's another complication we can introduce, where we say, okay, these two regions get this pattern of stimulus, and these two get a different one. They get these short pulses, and these have these long pulses. So let's see what that does to the brain. So we put it now into the variable stimulus. So all we have to do is go back and run our simulation again with this new stimulus. And let's see how that will look like as a plot here. And you can already see, oh my God, it's changing a lot. It's impacting the whole brain dynamics quite a lot, these two different stimuli now. And let's visualize the propagation again on this template brain. Let's wait a second. Yes, and you can see that now we have the stimulus starts for 4 different regions. And you can see that it really changes the activity nearly everywhere. So the more stimulation you give, the more excitation you will have in your system. That's quite, in this toy model, easily to trace. All right, so with that, we have finished this notebook, and we'll just shortly go back to the slides, where I just want to give you a couple of references. I think the most easiest to access one is our YouTube channel, Brain Modes from our lab, where you will have all kinds of videos explaining how to use the virtual brain, how to install the virtual brain, and what cool things to do with it. Then from our newspaper, actually all the code is online on GitHub. So you can also play with this multi-scale model if you like, to get some spikes out and some average activity on the population level. Yeah, and we also gave a much longer virtual course on exactly this topic of like 3 hours, where we took a lot of time explaining details and commenting on the code. So that's also on YouTube. Feel free to watch that as well. And there are always courses with the virtual brain from our team. Coming up again, virtual courses at this time, unfortunately. And then we have a large team, so feel free to contact any of us. Or Petra would be always best. She knows who has the overview still. And then I want to point out to Katarina, who should be actually there present with you guys. She just joined the team half a year ago. And feel free to also ask her, but I would also look forward to getting some Twitter messages or emails from you and hearing your reactions on this video or anything else you would like to discuss, maybe on deep brain stimulation. With that, I thank you very much for your attention, and I hope you're having a great day. So thanks. And yeah, let's stay in touch. Bye-bye.